Hello guys, now we are going to discuss about bacterial growth curve. Bacterial growth curve is one of the most important repeated question in microbiology 5 marks. Okay, now we are going to discuss about it. When the bacteria is inoculated in suitable liquid culture medium and incubated, the growth follow a defined course. It have 4 phases. First, lag phase, log phase, stationary phase, and decline phase. Here, in this growth chart, you can clearly appreciable in the lag phase there is a no growth of bacteria, because in the lag phase of the bacteria is not multiplying. At the same time, the size of the bacteria is increased and the bacteria synthesize enzymes and metabolized to multiplication of the bacteria. Okay, in the log phase the bacteria is multiplication starts. Here you can see, and in this log phase it utilizes the enzymes and metabolites to produce multiplication of the bacteria. In the stationary phase, there is a no multiplication of the bacteria. That means the completely stop the multiplication of the bacterium. And phase of the decline, the bacterium is completely stop the bacteria. And here you can see clearly appreciate in the log phase. That means the period between inoculation and beginning of multiplication of the bacteria. I already told. After inoculating into a culture medium, bacterium don't start multiplying immediately. It builds up enzymes and metabolites. In this log phase, the bacteria increase in size due to accumulation of the enzymes and metabolites. I already told. And bacteria reach maximum size at the end of the log phase. And coming to log phase. In this log phase, bacteria starts divide and smaller in size the bacteria are. In this log phase is the biochemically active phase, because this is the best stage to perform the biochemical reaction, and in this is the uniformly stained, and its the log phase is the best time to perform a gram stain. And coming to stationary phase. In the stationary phase, after the log phase, the bacterial growth ceases due to exhaustion of nutrition and accumulation of the toxins. Because when the bacteria multiplication starts, it utilizes the enzymes and metabolites, and finally it leads to accumulation of the toxin and the lack of the nutrition. Okay, and the number of progeny cells are formed. It's enough to replace the number of cells that die. Number of viable cells remain stationary, has almost a balance between dying and newly formed cells. The total count of the bacteria keeps rising. It's followed by bacterium becomes gram variable, more storage granules are formed, sporulation occurs. Bacterium produce exotoxin, antibiotics and bacteriocins. In the coming to last final phase, decline phase, gradually the bacteria stop dividing and completely cell death continues due to exhaustion of nutrition and accumulation of the toxins. Here you can see various phases of bacterial growth curve. Bacteria divides in lag phase? No, but it starts from log phase and stationary phase. In decline phase, the bacteria completely division is stopped. The bacterial death is In the lag phase, this is the starting phase to multiplication of the bacteria. So there is no death. In the log phase also there is no death. In the stationary phase, yes. Decline phase, definitely yes. The total count you can see in the log phase, or already you can already saw that picture, the flat. But in the log phase it's rise. In the stationary phase also rise. In decline phase is flat. And you can see viable count in the log phase is flat. In log phase it's rise, and stationary phase it's flat, and a decline phase it's falls. The special features between the lag, log, stationary and decline phases are: in the log phase you can see accumulation of the enzymes and metabolites. It attains maximum size. In the log phase you can see uniformly stained, metabolically active. The bacteriums are smaller in size. In the stationary phase, gram variable, it produces granules, spores, exotoxin, antibiotics, bacteriocide. In the decline phase, it produces involution forms. That's it, guys. Thank you. Subscribe for